All right, before we get started with our next biomolecule, let's do a quick review. Dehydration typically means that we need water. In this case, we are losing water. And synthesis means to build. So we are building something by losing water. Remember, water is the universal solvent. It is good at breaking things down. So we want to get water out of the picture to build stuff up. So when we look at glucose, which is a monomer of carbohydrates, and we are adding that to fructose, which is another of our three monomers of carbohydrates. We want to add them together. We essentially want to create a bond right here. And what happens is we are forming water to be lost as a product. And when we lose water, we are creating a bond here which links them together. So we are creating something more complex. Whenever you are building monomers into a chain, you are forming what's called a polymer. It is a very general term for just something more complex built in a chain for monomers. From a carbohydrate standpoint, since we have two different monosaccharides, in this case, glucose and fructose are monosaccharides, then sucrose would be known as a disaccharide. Reason being is it is made up of two sugars. Those sugars being glucose and fructose bonded together. So by losing water, we are making something more complex. Two things became one thing, more complex. The exact opposite of this is hydrolysis. Hydro meaning water, lysis meaning breakdown. So we are using water to its fullest advantage to break something down. So we have our sucrose, our disaccharide. Essentially, we're adding water to it. And this leaves us with our two monosaccharides. So one thing becoming more simpler because water is breaking it down. As you see here, sucrase. This is going to be an enzyme, which is actually a type of protein. We'll get to this later. Its whole purpose is to speed up the reaction. So, dehydration synthesis builds something up. Hydrolysis breaks something down. The key thing is when water is a reactant on the left side of the arrow, hydrolysis. Whenever we essentially see it, and we could write it like this, plus H2O. When we see it on the right side, and it is a product, then it is dehydration synthesis. So where is water? That'll help you figure out what is happening. All right, lipids. First thing with lipids, they are not polymers. We cannot form chains by adding monomer to monomer to form a polymer. The reason being is they lack a true monomer. We have completely different shapes among our three types of lipids that we're going to talk about. Because we have different shapes, they don't have that similar building block. So, what are they used for? First off, long-term energy. Similar to carbohydrates, they are an energy source. When you run out of carbs, you start to burn fat. Insulation. The more fat you have, the warmer you are. So a lot of animals that hibernate, they have to build up a fat layer to kind of last winter. And then, a lot of your animals that are more adapted to live in the cold, blubber and, polar bears have large fat layers to maintain body temperature in the cold weather. cell membrane is known as the lipid bilayer. What that means is there are two layers of lipids that form each cell membrane. And then chemical messengers, these are going to be your steroids, which also your hormones. I'm sure a lot of you have heard of anabolic steroids, which increase muscle mass. Steroids is a very general term. We'll talk about testosterone, we'll talk about estrogen, but for the most part, with our chemical messengers, we're going to talk about cholesterol and what cholesterol does to the cell. Similar to your carbohydrates, we do have C, H, and O, but there is no specific ratio here. Carbohydrates are 1-to-1. Lipids do not follow that. And then we do have one lipid that does have phosphorus. And then we don't have a specific building block because we lack a true monomer. So each lipid is different. So let's talk about the three different types. Our first type is called a triglyceride. Anytime you hear the word fats, we're referring to triglyceride. Okay, tri means three, and glyceride means glycerol. So what this stands for is we have what's called a glycerol head, kind of a base. Or glycerol group if you'd like. And then we have three fatty acids. That's where the tri comes in. That kind of stem off. So if you see a structure that looks like a capital E, that's going to be your triglyceride, lipid. Kind of easy thing to remember. Triglycerides are also probably what you come across on a daily basis when it comes to nutrition and food. Phospholipids. Let's break down this word. Phospho actually stands for phosphate, which contains phosphorus. This is where we get our P. Like I said, there was one type of lipid that does have phosphorus. This composes the cell membrane. Remember, we said it was the lipid bilayer. Bi meaning two layers of lipids. These lipids are kind of pointing opposite to one another. So we have the phosphate head, and it is polar. And since water is polar, it's called hydrophilic. And philic means water-loving. So it wants to be near water. So you have your phosphate heads pointing outside the cell as well as inside the cell. So you have water that can come up to both sides. And then you have two fatty acid tails, similar to what we saw with the triglyceride, but now there's two. And these are nonpolar. And since polar and nonpolar do not get along, these are hydrophobic, which means they are water-fearing. So nonpolar everywhere in the middle, polar everywhere outside and inside. So it creates a nice barrier to keep things out and to keep things in. And later on we'll talk about how this specific structure allows the cell to kind of maintain homeostasis when it comes to things coming in and out, whether it's water or other nutrients that the cell needs. All right, the final thing is steroids. Very general term. Your hormones, chemical messengers, testosterone, estrogen. Testosterone, typically you think of your males, estrogen, your females. in terms of hormones with kind of how we look the way we look. We're going to focus on cholesterol. Cholesterol, you think, may cause heart attacks if it builds up, in your arteries. We're talking at the cellular level here. So cholesterol at the cellular level, within the cell membrane, they're almost like anchors. Your cell membrane is very fluid. It kind of moves. So to kind of keep things together, we have cholesterol kind of throughout the cell membrane just to kind of keep it intact. Now, final thing about lipids. Remember, this is our triglyceride. We have our glycerol. And of course our 3 fatty acids. Now, one thing to look at here is we have a difference right here. We have a double bond. This fatty acid is going to be known as an unsaturated fatty acid. And these two, since they are all single bonds, are going to be saturated fatty acids. Since one of the fatty acids is unsaturated, this entire triglyceride will be known as unsaturated. Now, why this is important is your saturated fats only have single bonds. So since they are very symmetrical, there's no weak link, and that's why they appear as solids. Your unsaturated fats are going to have a double bond somewhere, which causes that kink or that bend in the structure that we see right here. That creates a weak spot, and that's going to change the phase of matter that the fat is in. So you're going to see your liquids here. So your olive oil, your vegetable oil are unsaturated. Because these are in liquid form, because we have that distinctive kink, which leads to a completely different appearance, these are going to be easier to digest and break down. So as a result, your unsaturated fats are going to be healthier. Now, olive oil is healthier to use than butter. It's all due to that double bond that is within the triglyceride. Now, final thing, just quick review, carbs versus lipids. Things they have in common. They both are involved with energy. They both are involved with structure. And they both contain carbon, hydrogen, and oxygen. Major differences. Carbohydrates are going to be your primary source of energy. It's going to be quick and kind of a fast way to access energy right away. Lipids are going to be more your secondary source because they're more long-term storage of energy. From a structural standpoint, we have carbohydrates make up the cell walls of plant cells, whereas lipids make up the cell membrane. And this is going to be for plant and animal cells. So our barriers. And finally, we have a 1-to-1 ratio of CH and O of carbohydrates. An example would be glucose. Glucose C6H12O6. That 1-to-1 ratio. There is no ratio with lipids. So if you only have carbohydrates and oxygen, count them out. If it doesn't follow that ratio, then it is a lipid. And also, your phospholipids do contain phosphorus. And this is what leads it to be polar. So think phosphorus polar when it comes to your phospholipids. The final big distinction is going to be your monomer. Carbs have one. It is called a monosaccharide. Remember, mono means one, saccharide means sugar. So one sugar, because we can bond them up to form carbohydrate chains. This lacks a true monomer, because we have three different shapes of lipids. You have your triglyceride, you have your phospholipid, and you have your steroids. So a whole bunch of different shapes, which means there's not a building up of a chain similar to your carbohydrates. Okay, that's it for lipids. If you have any questions, please ask. Thanks for watching.